For decades, the study of human cognition has been trapped inside loud magnetic tubes. On the left, traditional functional MRI, and noisy, blurry approximation of localized blood flow. On the right, a pristine computational prediction generated entirely in software. The physical fMRI machine is the historical bottleneck of neuroscience. It forces researchers to observe the brain under highly artificial constraints. Subjects lie perfectly still, reacting to isolated single-sensory tasks, like looking at a static image or listening to a lone beep. The data gathered in these machines is heavily corrupted by the human body itself. Every heartbeat, every respiratory shift, a subtle twitch, or a wandering attention span introduces severe physiological noise into the raw imaging data. Attempting to scan subjects experiencing natural, chaotic reality, like watching a movie or listening to a podcast, amplifies this noise. Acquiring clean, multimodal data across these complex environments is financially prohibitive and highly susceptible to distortion. Due to these physical constraints, cognitive neuroscience has struggled to build a unified model of human thought. The field has been forced into isolated subdisciplines, analyzing discrete visual or auditory functions, rather than mapping how the brain synthesizes the messy, multimodal reality of everyday life. Meta's fundamental AI research team has built a solution. It is called the Transformer for In-Silico Brain Experiments, version 2, or TRIBE v2. It is a deep multimodal predictive foundation model designed to operate as a high-fidelity digital twin of human neural activity. Where early computational models could only predict a coarse approximation across roughly 1,000 localized brain parcels, TRIBE v2 scales this output by a factor of 70. It predicts time-resolved synthetic brain activity across 70,000 distinct voxels, covering the entire cortical surface and deep subcortical structures. To achieve this resolution, Meta trained the architecture on an immense dataset, over 1,115 hours of high-resolution fMRI recordings. This data comes from more than 720 healthy human subjects who were exposed to a rich diet of naturalistic media, including movies, podcasts, and text. This is a predictive engine endowed with robust zero-shot generalization capabilities. It can accurately predict the spatially resolved hemodynamic response for entirely novel subjects, unseen languages, and new experimental tasks, without requiring any fine-tuning or access to a physical scanner. By decoupling neural experimentation from the physical hardware, TRIBE v2 operationalizes In-Silico Neuroscience. Researchers can now simulate, test, and iterate complex neurocognitive hypotheses entirely in a computational environment, processing thousands of simulated brain experiments in seconds. Simulating human perception requires translating 3 distinct sensory inputs: language, acoustic sound, and dynamic video, into a unified mathematical prediction of localized blood flow. The TRIBE v2 architecture relies on 3 generative AI foundation models, frozen as feature extractors. Text passes through Llama 3.2, audio through Wav2Vec BERT, and video through V-JEPA 2. These neural networks exhibit representational geometries homologous to the human brain, establishing a precise baseline before fusing modalities. The raw embeddings derived from those 3 separate modalities reside in completely disparate mathematical latent spaces. To unify them, the architecture compresses and concatenates each stream into a standardized 1,152-dimensional continuous multimodal time series. This diagram shows the core learning engine of the architecture, an advanced 8-layer temporal transformer block. The temporal dynamics of this transformer are engineered specifically to emulate human biology. It processes the aggregated data streams across an expansive 100-second context window. The fMRI signal the model predicts is driven by the hemodynamic response function, or HRF. This is an indirect, metabolically sluggish measure of neuronal firing. When a neuron fires, it takes 5 to 6 seconds for localized blood flow to peak and up to 30 seconds to return to baseline. The 100-second temporal aperture grants the transformer the capacity to mirror this slow physiological delay. Through its self-attention mechanisms, it learns how narrative tension built over 40 seconds naturally shapes the neurovascular reaction to a specific visual or auditory stimulus occurring at second 41. During training, the researchers apply a strict regularization technique called modality dropout. Each of the 3 input streams is intentionally zeroed out with a 30% probability, forcing the network to maintain predictive accuracy even when a specific sense is missing. Modality dropout compels the model to abstract away from purely surface-level sensory correlations and build deep, amodal semantic representations. This mathematically mirrors the function of high-level associative cortices, like the temporal parietal junction, where sights, sounds, and language are fused into a singular human experience. The final architectural challenge is anatomical. Every human brain exhibits unique structural folding patterns of gyri and sulci, making the translation of universally integrated latents into precise physical voxels highly complex. Tribe V2 manages structural variance through a subject block, fine-tuning the mathematical projection for the exact individual. To prevent overfitting, subject dropout bypasses this block 10% of the time. Representations are forced through an unseen subject layer, mathematically formalizing an idealized, canonical neurocognitive response. By aggressively regularizing across hundreds of individuals, the AI successfully filters out the physiological noise inherent to physical data collection. This raises a counterintuitive hypothesis. Could a software model become a cleaner biological sensor than the actual human body? This chart shows Tribe V2's accuracy on the 7 Tesla human connectome dataset. Achieving a 4.0x zero-shot score, it doubles the predictive accuracy of the median human scan. By stripping away biological noise, the model estimates population-level neural activity more accurately than physical biology allows. If a foundation model can perfectly predict localized brain activation from a known sensory stimulus, the next logical step is mathematically inverting the process. This flowchart illustrates simulation-based inference, decoding synthetic brain maps to reverse engineer stimulus parameters. A summary backbone compresses the 20,484 vertex mesh, while a generative network deduces original input. Testing successfully recovered exact emotional parameters, valence, arousal, and dominance from text-prompted synthetic maps. When researchers fed the forward-backward pipeline with unprompted real-world headlines from prominent global news outlets, the inverted model successfully differentiated deep stylistic and emotional variations across publishers, operating entirely by interpreting simulated topographical signatures. The successful deployment of simulation-based inference transforms foundation brain models from passive predictive tools into active generative decoding engines. Running this biological loop in reverse introduces the capability for algorithmic inverse stimulus design. In commercial applications, programmatic neuromarketing allows systems to computationally search massive design spaces. Media engineers can synthetically generate audio tracks or video cuts mathematically guaranteed to target and induce specific predefined neural states in a human audience without ever surveying a single test subject. Autonomous humanoid robotics is the highest stakes application. Embedding Tribe creates human digital DNA, granting robots mathematically rigorous empathy to anticipate human fatigue in high-stress environments. Deep neural networks have evolved from statistical computational tools into mechanistically accurate structural analogs of biological cognitive processing. Tribe V2 marks the arrival of In-Silico Neuroscience. Researchers now possess the mathematical architecture to observe how the human brain passively reacts to reality, and the inverted computational power to systematically engineer the sensory